In this segment of the course, we're going to begin to talk about biological approaches to treatment of schizophrenia. We've talked a little bit already about the neuroscience of the disorder, about some of the changes in the brain that are often associated with the diagnosis of schizophrenia. And what I'll be doing today is focusing more on looking at what medications are most effective in the reduction of symptoms of schizophrenia. It's clear that the first line of treatment for symptoms of schizophrenia is the use of antipsychotic medications. Antipsychotic medications treat two major aspects of the disorder of schizophrenia. They treat or affect delusions, and they have an influence and can affect hallucinations as well. So those two positive symptoms, what we've described before in the course as being examples of positive symptoms, are affected by antipsychotic medication. It's important to also know what aspects of the disorder are not affected by antipsychotic medication. That is to say, the negative symptoms that we've talked about, the cognitive difficulties that we've talked about, the social cognitive difficulties that we've talked about in the disorder, they are resistant or tend to be much less responsive to the effects of antipsychotic medication than are the positive symptoms of delusions and hallucinations. So, as should be clear from this description, the appropriate treatment of people with schizophrenia involves a comprehensive approach in which both medications are utilized, antipsychotic medications are utilized, but very carefully in coordination with a variety of psychosocial interventions that we will be talking about in the course, to really try and attempt to treat the entire client with the disorder and address the vast array of difficulties that people with schizophrenia may experience. So in terms of biological treatments, we can talk about two major classes of antipsychotic medication. One major class is what's been called the first generation antipsychotic medications. And these relate to the first antipsychotic medications that were discovered for the use for schizophrenia. And then a second generation of antipsychotic medications that emerged largely in the 1980s that also are designed to address the delusions, hallucinations that people with the disorder often experience. As we talked about in a previous segment, it has been shown in a variety of studies that antipsychotic medications appear to have their effect by affecting a substance in the brain known as dopamine. Dopamine is one of a group of 20 or even more brain chemicals that are involved in signaling between different neurons. And again, neurons are the sort of elementary units that make up the brain, right? The sort of elementary functioning units that make up the brain. We've talked about the first generation antipsychotics and their important role in blocking D2 receptors in the brain. And specifically, they appear to target D2 receptors, which are located largely in what we've talked about as being the substantia nigra projection, or the nigrostriatal projection, and in the mesolimbic dopamine system. And that these drugs appear to block dopamine 2 receptors, making it more difficult for dopamine to signal between two neurons. And this may be the mechanism by which delusions and hallucinations are decreased in the disorder. Now, at the same time, some questions came up about this sort of biological mechanism for treatment with the emergence of what have been called the second generation antipsychotic medications. The second generation antipsychotic medications are characterized by reducing positive symptoms. So just like the first generation, they are effective at reducing hallucinations and delusions. But their mechanism of action appears to be quite different. That is to say, they don't have the same affinity or attraction to dopamine 2 receptors. The best example of this type of a drug would be clozaril, or clozapine. Clozapine is a drug that was rediscovered in the 1980s. It had been used prior to that, but in the 1980s was used in a major randomized controlled trial. And in this randomized controlled trial, they took people with schizophrenia who had had poor responses to 3 different types of antipsychotic meds. So these are folks who really were non-responsive to antipsychotic medication. And they took these folks who were non-responsive to 3 antipsychotic meds and randomly assigned them to receive either chlorpromazine for 6 weeks or this rediscovered drug, or this newer drug, known as clozapine. And what they found is in these very hard-to-treat clients, these clients who really had just shown very little response to many of the medications, that 30% of the folks that were in the clozaril condition actually showed significant improvement in their symptoms. This contrasted with 4% in the chlorpromazine group. This was interesting for a number of reasons. For one reason, it was important because it showed that you could have a medication that was effective for people who seemed to do poorly on many different medications. That is, it seemed to be a drug that might be effective for people who just didn't respond to other medications. So that was exciting and a real innovation, number one. But number two, the other thing that was interesting about this drug, clozapine, is that it had not such a high attraction to these D2 receptors that we talked about as being so crucial for the antipsychotic effects of the first-generation antipsychotic medications. Instead, these drugs tended to block receptors for other neurotransmitter systems like serotonin, for acetylcholine, which is another neurotransmitter system, and they seem to act on histamine receptors in the brain as well. So the success of clozapine as a medication led to the development of a variety of new second-generation antipsychotic medications that were not necessarily characterized by a high affinity for these D2 receptors. This has led to questions about which of the drugs may be more effective. Are the second-generation antipsychotics most effective? Are the first-generation antipsychotics most effective? And what does it mean that the second-generation antipsychotics seem to promote their effect without necessarily blocking these D2 receptors, or at least not at the same ratio or with the same intensity as the first generation? Recently, a study was conducted called the CATIE trial, which is the clinical antipsychotic trial of intervention effectiveness, in which they looked at over 1,400 individuals with chronic schizophrenia at a total of 57 clinical sites. In this study, they compared 4 of the newer second-generation antipsychotics with 1 of the old first-generation antipsychotics that had been around since the 1960s. The question here was, which of these medications was more effective for people with the diagnosis of schizophrenia? Their primary outcome in this study was how long clients stayed on the drug, and they looked at other outcomes such as symptoms and functioning. The important thing to recognize from this study is that for most of the outcomes that were looked at, there was very little difference between, on the one hand, the second-generation antipsychotics, the 4 that were looked at, and on the other, the first-generation antipsychotics. So despite a increase in interest in prescription patterns that were associated with the advent of the second-generation antipsychotics, what the data suggested from this trial is there wasn't a whole lot of difference in the effectiveness of the old antipsychotics as compared to the new antipsychotics. And this, of course, is very important when we think about the cost of these medications, etc. The last idea that I would like to share with you is the most recent data that really suggests, in a much even stronger way than what I've presented to you so far, that the effects of these antipsychotic medications appear to be through their blockade of these postsynaptic dopamine 2 receptors. These are studies that have used a technology called PET, positron emission tomography, and like fMRI, which we've already talked about, it measures activity of the brain. It's not measuring structure, but rather activity of the brain. In these PET studies, there are intravenous infusions of a molecule that is radio-labeled, that it is radioactively labeled. It is infused into the blood, and this molecule is selective to bind to those postsynaptic protein receptors that we talked about earlier. So these dopaminergic postsynaptic receptors are bound to by this chemical that we introduce into the bloodstream that is radioactively labeled. This allows us to create an image by the radioactivity that is emitted of the areas in the brain that have the highest density of these D2 receptors. These studies have revealed several interesting findings. One interesting finding is that in people with schizophrenia who have been administered these radio-labeled molecules, these radio-labeled molecules that are located specifically in the striatum, the degree to which these radio-labeled molecules are displaced gives us a sense of how many receptors there are or how much dopamine activity occurs. That is to say, if dopamine is stimulated in the striatum, we would expect larger amounts of this radio-labeled chemical to be removed from the receptor sites because they get displaced by the dopamine which is being transmitted. In these studies, they found that when you administer a drug that increases dopamine levels, people who have higher symptoms, higher levels of delusions and hallucinations, show more displacement of the radioactively labeled molecule, suggesting that these increases in symptoms that are associated with a challenge or an increase in the amount of dopamine that's transmitted in the brain relates directly to the increase in symptoms that you see in these folks. Another way that people have tried to look at this is to look at folks just at baseline without any type of stimulation of dopaminergic activity and just ask the question, to what degree do these radio-labeled receptors, to what degree are they occupied by these molecules at rest when we decrease the amount of dopamine that's being transmitted? And sure enough, people with schizophrenia with higher numbers of symptoms or higher levels of symptoms tend to show higher levels of radioactivity at these postsynaptic receptors, again suggesting that there may be more activity or there just may be the presence of more dopaminergic receptors in people with schizophrenia. These findings are very important because they are occurring in the living brain. These are in people with schizophrenia who are obviously suffering from the disease, and you are actually measuring the levels of dopamine transmission in the living active brain. So for this reason, they've been thought to be very, very important.